Okay, well, good morning, everyone, and welcome to Cowen's 2019 Healthcare Conference. I'm Irina Margina. I'm on the Biotech Equity Research team, and it is my pleasure to introduce this morning Lou Brenner, who's the CEO of Olema Pharmaceuticals.
Thank you, Irina, and good morning. Thanks to Cowen for hosting this conference and for inviting us. As a reminder, I will be making forward-looking statements this morning. For more detail on these, please refer to our SEC filings, including our recently filed 10-K. First, I'd like to take a moment to acknowledge our co-founding CEO, former CEO Alexey Margolin, who really helped build this company and gave me a chance to succeed him in this role that I'm quite grateful for the opportunity to lead Olema to the next phase. What we are doing in our company is to build a platform of oral enzyme therapies that work in the gut. They do this in order to hit targets that are associated with metabolic diseases that affect the kidney and other organs. We have made significant progress with our platform, including our lead asset, raloxolaiase. This is a phase 3 compound in development to treat severe forms of a disease called hyperoxaluria, which refers to too much oxalate in the urine that can be damaging to the kidneys. Earlier this year, we made an important announcement of an acknowledgement of our alignment with the FDA for our phase 3 program and a strategy to pursue accelerated approval. I will go into that into detail later in the presentation. We are presently running 2 phase 3 studies called UREROX-1, for which early data is expected at the second half of this year, as well as recently starting UREROX-2. Our technology really allows us to be able to treat metabolic diseases via the gut using a GI mechanism of action. We also have a second product candidate that is making its way through the late phases of preclinical development, and this will be called ALN-346. It is designed to treat hyperuricemia or gout in patients with chronic kidney disease. We believe that these unmet needs in both oxalate and urate disorders would position us well to serve patient populations that currently are lacking available or sufficient therapies. The big announcement that was made by the company, and it represents a culmination of a prolonged period of constructive negotiation with the FDA, led us to announce the accelerated approval designation, or the accelerated approval potential path for our lead program in raloxolaiase. This is a compound that is trying to reduce urinary oxalate. Urinary oxalate is a marker for progressive disease in these patient populations. The UREROX-1 study, which has been running since last year, and the UREROX-2 study will have the same primary endpoint, which is a % change from baseline in 24-hour urinary oxalate excretion. The UREROX-2 study will have a long-term endpoint associated with kidney stone disease progression and also has an adaptive design element. The common features of these 2 studies are important in that the first study will allow us to understand better the likely outcomes of the second study, and taken together this will represent the most robust program that has been conducted in oxalate disorders. Importantly, the FDA process for this discussion was quite data-driven, quite collaborative, and we believe led us to quite an attractive and important place to try to help patients who currently have no available therapies. Our pipeline is focused on, at first, treating patients with enteric hyperoxaluria. This is a severe form of the disease associated with gut disorders that I'll go through the physiology in a moment. We also have orphan designations in the rare genetic disease of primary hyperoxaluria as well as pediatric hyperoxaluria, and we are running a basket study to treat these patient populations called the 206 study that I will detail at the end of the presentation as well. And as I mentioned earlier, we are progressing through preclinical development for ALN-346 for the treatment of hyperuricemia and chronic kidney disease. Our technology platform is based upon oral enzyme therapeutics that we are able to design, manufacture at scale via fermentation, formulate for activity in the GI tract, and then deliver to hit a known set of targets in the GI tract. We are using this to develop first-in-class enzyme therapeutic candidates, including raloxolaiase and ALN-346. These classes of therapies have the ability to potentially make their treatment in the gut without absorption of the enzyme. We have made significant progress in our scalability of manufacturing, including both the yield and the activity of the enzymes. If you think about therapeutic classes that are relatively similar, although none have been exactly similar, you're thinking about sequestrants like bile acid sequestrants, binders, including polymers to bind phosphate and potassium, and then you're thinking about enzyme replacement for diseases like cystic fibrosis and pancreatic insufficiency, or as well as a milk sugar allergy using lactate. But those are the therapeutic classes. There's not been yet a bacterial-based enzyme to treat the therapy in the gut, and we hope to bring these to the market for the first time. A bit more detail on oxalate. Oxalate is a 2-carbon carboxylic acid that is ever present in plant material, as well as a result of metabolic activity in the body from carbohydrate as well as amino acid metabolism. When you eat oxalate, or when you eat vegetables and plants, you liberate oxalate because it is used to store calcium stably. As oxalate comes into the body from food, it is liberated and absorbed through the GI tract. Once oxalate enters the body, it has to leave via the kidney. There is obligate filtration and secretion through the kidney to eliminate oxalate. In normal physiology, this is usually not problematic. However, in settings of excess oxalate, the kidney and calcium levels of oxalate become overwhelming, and you can see precipitation of crystals and ultimately stones. The other pathway for oxalate generation is from the liver, where a variety of liver enzymes result in oxalate from normal carbohydrate as well as amino acid metabolism. In the rare genetic diseases of primary hyperoxaluria, there are enzyme deficiencies that lead to excess generation by the liver. The kidney does not know the source of oxalate, whether it's metabolically generated or from the diet. In the enteric conditions which we are focusing on, these patients have hyperabsorption of oxalate due to an underlying GI disorder, either associated with malabsorption or inflammation. And with that, you see excess levels of oxalate that the kidney must handle, leading ultimately to kidney stones. In the most extreme form of the disease, you can actually get kidney failure, and there are patients who have come to dialysis as well as transplant because of this condition. When you think about kidney stone disease, as well as hyperoxaluria as a potential cause of kidney stone disease is quite common. 10% of the U.S. population will have a kidney stone at some point in their lives. About half of those will have a recurrence. Hence, you see the bottom of the pyramid with significant millions of patients having idiopathic kidney stone disease. Our focus has been on the high-risk middle of the pyramid here in blue, the enteric population. Enteric conditions refer to malabsorptive or inflammatory conditions of the GI tract, including surgical conditions like gastric bypass surgery as well as short bowel syndrome, and then medical conditions including inflammatory bowel disease like Crohn's disease, ulcerative colitis, and then disorders of malabsorption like celiac disease, pancreatic insufficiency, and liver disease. In these settings, the absorption of oxalate becomes excess. These patients have a significant burden of kidney stone disease and are at risk of kidney failure. So as we peel back this 250,000 patients that we've estimated, we see as we've looked at them in our phase 2 program that on average they've had 6 kidney stones at some point in their lives before entering the trial. And you can see the scan here that these patients had on average 3 kidney stones in their body at the time of enrollment. So for these patients, kidney stones are not just a one-time occurrence, but they are really a significant impact on their overall health outlook. And as you probably know, they can be quite painful and cause significant loss of function due to the chronic issues associated with kidney stone disease. As we think about urinary oxalate levels, they are assessed over 24 hours. The normal urinary oxalate excretion is up to around 30 or 35 milligrams a day. In normal physiology, that's around half of it metabolically generated and half of it from the diet. However, in a series of different conditions, you can have excess from either of those cases. For instance, in primary hyperoxaluria, the metabolic generation becomes excess, leading those patients to have more than 100 milligrams a day of oxalate excretion. And you can see on the y-axis, as the oxalate levels go up, the risk of kidney stones, kidney damage, and ultimately kidney failure becomes significant. In the setting of enteric hyperoxaluria, you can also see the oxalate levels rise, but in that case, most of the oxalate is due to GI malabsorption. And with this, you can see levels in enteric hyperoxaluria that approach the levels in primary hyperoxaluria. In fact, in the enrolled population in our phase 2 study, we saw approximately 100 milligrams a day of oxalate excretion in our patient population on average. The goal therapeutically, as represented by the arrow, is to reduce the daily burden of oxalate on the kidney. There are a series of publications listed here on the right from the American Society of Nephrology, from the New England Journal of Medicine, from Kidney International, all that suggests that reduction of oxalate epidemiologically should be associated with long-term benefit in these patient populations. From this, there is a solid data-driven estimate of a 20% reduction in urinary oxalate as being a clinically meaningful endpoint, and this you will see as our secondary endpoint in our phase 3 trials. We've also looked at this patient population from the top down, based upon risk and following this patient population in a payer database using the IBM TrueVan MarketScan data. As we think about the in silico risk of these patients as they have been followed for a 4-year period, you see around 4.4 million patients who have had an initial diagnosis associated with a GI condition that would lead them to risk, but no prior diagnosis of kidney stones or kidney damage. As this patient population is followed over 4 years, around 5% will develop their first kidney stone, and beyond that, another 2% will develop chronic kidney disease as well as kidney stones. And at the far end on the right, some patients from this condition will develop kidney failure. So you can see in a relatively short period of time that these conditions of a first diagnosis of a GI condition of risk will lead to a significant risk of both kidney stone disease and ultimately potentially kidney failure. As we think about the size of the population in the second level here, the 250,000 is an estimate that we've reached from both this top-down claims analysis as well as the bottoms-up disease-by-disease analysis. We believe this is important because this market has not been characterized in the past. This is a market where many physicians have seen these patients, but there's really been nothing, no therapeutic, no consistent approach to unify this market. And this is one of the opportunities that we believe that we will have to help this underserved patient population. We are not alone in the effort to develop new therapeutics for hyperoxaluria. And as I take a broader view of what's happened in the industry over the last several years, as the collaboration led by the Oxalosis Foundation here in the United States, as well as Oxal Europe, and then in collaboration with the Kidney Health Initiative and the American Society of Nephrology and the FDA, have all been working to identify different ways to develop new products for patients with oxalate disorders. With this, there is a Kidney Health Initiative white paper that's being led by the Mayo Clinic that is currently being produced in order to define endpoints and trial designs for patients with oxalate disorders, including primary as well as secondary hyperoxaluria and enteric hyperoxaluria. These patients have initial presentation of recurrent kidney stones in most cases. That leads to the diagnosis of this condition. And ultimately, as you can see on the far right, there are patients in the United States every year who go on to dialysis because of oxalate disorders, including enteric hyperoxaluria. I'll now go into the data, looking at raloxolase and the work that's been done for the treatment of hyperoxaluria. As we've laid out here, the development has been progressive, with learning at each and every stage of drug development, starting with a healthy volunteer study in the secondary hyperoxaluria that paved the way for a phase 2A open label study, and then randomized phase 2 program that I'll cover, especially the 713 study that was presented at the ASN in 2017, ultimately leading to our 2 ongoing phase 3 studies, UROX-1 and UROX-2, the substance of which could lead to an accelerated approval filing and ultimately approval for a first-in-class therapy to treat patients with enteric hyperoxaluria. I'll also call your attention to the bottom line, are the patients with primary hyperoxaluria, for which we have orphan designation. We have preclinical data that's published to support the potential mechanism of action in this orphan disease population, even though we acknowledge that enteric hyperoxaluria is a better match mechanism of action to a mechanism of disease in the GI tract. But we do think there is solid rationale for looking at patients with primary hyperoxaluria, even though this program is somewhat earlier in its evolution. We are running the 206 study, which is a basket trial in this population, ongoing. The clinicaltrials.gov references are all here. I'll also call your attention to the fact that our strategy has been to run phase 2 open label studies. On the top, which was originally the study presented at AS in 2015, we're similarly running a signal-seeking open label trial in the most severe forms of the disease at present. Our 713 study was the most important study that we've run so far in the phase 2 program. This was a randomized phase 2 study involving 67 patients overall, with a pre-specified look at both the idiopathic population as well as the subset of patients with enteric disorders represented in the purple. In these studies, we see a consistent effect of urinary oxalate to reduce the excretion of oxalate by varying degrees in different patient population. The primary endpoint was change in urinary oxalate in the overall population at week 4. We also had pre-specified analyses for reduction in the enteric population, looking also across the study in an area under the curve or time-weighted average. You see here on the right the effect size of 25 milligrams for 24 hours, or up to 39% compared to placebo, is more significant in the enteric population, and that the data in the time-weighted average across 4 weeks is more consistent, given that it allows for more measurement over the course of the study rather than a single time point at the end of the study. You also see in the bottom, as we take thresholds or cut points for different degrees of reduction compared to placebo, whether it be 10%, 20%, 30%, 40%, or 50%, there is a separation mathematically relative from raloxifene relative to placebo. You'll also call attention to the second column in the bottom, the 20% threshold showing a difference of 64% versus 14% raloxifene versus placebo. Based upon this data, we have carried forward the enteric population into phase 3. We've also carried forward the aggregated endpoint over 4 weeks, average data looking at an area under the curve analysis as the primary endpoint. When you look at the data in terms of a scatter plot and a bar graph, you think about on the left that the effect is most significant in the enteric population and really a driver of the overall population. Hence our decision based upon a higher unmet need as well as a bigger effect size to carry forward the enteric population. We do think there will potentially be strategies to go back to the secondary population or idiopathic based upon some patients being more likely to hyperabsorb. And with that, we have run a study called the 204 study looking at oxalate challenge tests, where we fed patients a spinach souffle and allowed them to see their levels of oxalate absorption. So we do think the overall population or secondary hyperoxaluria is one that we could approach again with time and resources in the future. I'll call your attention to the figure on the right, the scatter plot, and really we're thinking about in our phase 3 studies comparing the triangles, those patients with enteric hyperoxaluria, to the open circles. With this, we see that more patients, or the majority of patients on therapy, do show a negative change or below the line compared to the placebo patients, which you see some patients above and some below, but the net effect of placebo appears to be relatively flat, as shown on the left. I'll also call your attention to those few patients right around 50 milligrams at entry. We have modified the process for enrollment in the phase 3 compared to the phase 2, where patients will need to have an average of at least 50 and no value below 40 at the time of enrollment. And so some of those patients to the left end of the figure at around 50, where the effect size has not been as significant, would be unlikely to enroll in the future study. So we think that this is a data-driven strategy to try to really enrich for the phase 3 population. In terms of safety, the drug has been well tolerated so far. It is an oral non-absorbed protein, and with that there may be some degree of GI side effects. We've not seen any difference from the active versus the placebo across our studies. We have exposed patients so far in randomized clinical trials for 1 month at this point. So as we summarize the learnings from phase 2 before we go into phase 3, we know that enteric hyperoxaluria is a rare disease without an available treatment. We are focusing on percent change in urinary oxalate. We have a secondary endpoint of 20% reduction. We have increased the dose, and this is a point that I haven't made yet, but I think is a really important one. We learned that in prior studies that patients were, on average, some patients were consuming about 20 to 30% of their daily oxalate load in a period when we weren't dosing medication. We are increasing the dosing frequency from 3 to up to 5 times per day, and this we believe will give the potential for more oxalate degradation in the phase 3 study. We also have a 6-month follow-up in the urinary oxalate biomarker in the second study, or UROX2, and importantly at the bottom we have designed a kidney stone disease progression composite endpoint, as well as long-term measures of kidney function, as well as resource utilization associated with kidney stone disease as the long-term endpoint in the 302 or UROX2 study. This is probably the most important slide of the presentation, slide 18, where we're focusing on the 301 study and progress that's been made. As I just mentioned on the left, the key design elements of the phase 2 study is that we have tried to reduce the variability of urinary oxalate, which was one of the key challenges that we identified in the phase 2 program. This is a biomarker that's not been used for clinical development thus far, and in our statistical work and in our discussions with the FDA, finding ways to reduce variability has been quite important. As I mentioned earlier, we've changed the strategy for baseline setting, where patients need to have both a screening as well as a baseline level that would qualify them for the study in an effort to keep the average above 50. We've also enhanced site training. And then I mentioned the bottom figure here, the bottom bullet, that we increased the dose frequency to better match the eating patterns. So how are we doing? In terms of progress, the study is more than 50% enrolled. We have looked at pre-treatment data or the screening and enrollment data on patients in the initial patient cohort of that first 50% or so of patients, and we've seen that there is decreased variability, in fact, in urinary oxalate compared to that which we observed at the prior phase 2 study, including the 713 study, showing that this effect that we've tried to change the process of screening and baseline has indeed led to a reduced variability in urinary oxalate. As you probably know, in terms of the statistical calculation of success, there's the effect size, and then there's the standard deviation or the variability. And so we believe that by reducing the variability, we have the potential to really show the effect size more significantly. We've identified no safety concerns so far in the blinded data, and the study is on target to deliver top-line data in the second half of 2019. And as I mentioned before, the primary endpoint being this change in urinary oxalate measured over 4 weeks, which is also the primary endpoint for the URIROX-2 study. Our update on URIROX-2 is that the study is similar in design in that it uses the same primary endpoint, the urinary oxalate biomarker, and the same secondary endpoint of a 20% reduction. The patient population is essentially the same, although we do require that patients have a history of kidney stones, which will not be a major challenge, but given that this is the long-term endpoint of the study. And we've stratified for patients who are either on bariatric surgery, which is a very common cause, or other enteric conditions. This trial has an adaptive design strategy where we will use the data from the first month, being the primary endpoint, and the 6 months of data on safety as well as efficacy to be able to do a power calculation and an adaptive design that will allow for the post-approval phase or the post-filing phase of the study to potentially deliver on the long-term endpoint, which is kidney stone disease progression. So as we summarize these two studies together, if you look, they have a common endpoint, URIROX-1 and URIROX-2, using the urinary oxalate surrogate marker. The primary endpoint is the percent change in urinary oxalate measured over 4 weeks. The lead secondary endpoint being the 20% reduction, and the dose increase of up to 5 milligrams per day, 5 doses per day compared to 3 doses per day, we believe is significant in that it will increase the potential for oxalate degradation by the enzyme in these patients. In terms of our progress as listed below, we expect to deliver our first phase 3 top-line data in the second half of this year. And as I mentioned before, that trial is tracking well with enrollment progress more than 50%. In addition, URIROX-2 was kicked off at the end of last year, and the process of site opening and screening is now well underway. In terms of additional indications, we're also interested in those patients with severe forms of oxalate disorders, including those patients who develop what's called hyperoxalemia, or excess oxalate in the blood. Your oxalate is a uremic toxin in that it is damaging to the kidney itself, and then as the kidney becomes further damaged, the amount of oxalate in the body increases because the kidney no longer has the ability to get rid of it. It's very similar in that way to urate, in that it can both damage the kidney and actually result from kidney damage. In this vicious cycle, you start to see oxalate build up in the body, a condition called hyperoxalemia, and then you can also see it what's called systemic oxalosis. As we think about our patient population, we're looking at patients realistically with primary hyperoxaluria who have a severe risk of kidney disease, as well as hyperoxalemia in this patient population. And some of these patients even will come to dialysis and be on transplant. In this setting, we're running the 206 study. This is a basket study looking at a variety of severe patient populations currently who have a very high unmet need. Our rationale is laid out here on the left. This is a signal-seeking study in multiple subpopulations. These include enteric patients with chronic kidney disease not yet on dialysis, enteric patients who are actually on dialysis, and enteric patients who have had kidney transplant, as well as patients with primary hyperoxaluria types 1 through 3. This is the first time we are assessing plasma oxalate to determine its utility as a subsequent endpoint in randomized controlled trials. This is much like we started by using urinary oxalate, which had not been used for regulated drug development, but we are investigating the use of plasma oxalate. This is used clinically at some centers, but not widely, and we're trying really to industrialize this for further use. We've also will have in this study our first exposure in patients on dialysis and patients with primary hyperoxaluria in adolescents. The hypothesis is that decline in kidney function leads to oxalate absorption, and that by degrading oxalate in the GI tract, you can actually pull it through the body and reduce the burden of oxalate systemically. The trial design is an open-label study of patients greater than 12 years old with either primary hyperoxaluria or enteric hyperoxaluria with hyperoxalemia. The key endpoint is change in plasma oxalate as well as 24-hour urinary oxalate excretion. As I mentioned before, plasma oxalate has not been used thus far for regulated drug development, and the 24-hour urine oxalate may be variable in these patients because their kidney function is reduced. For instance, patients on dialysis, you can't use that as a marker. In terms of the study update, we've enrolled 8 subjects across multiple primary and secondary or enteric populations. Our pretreatment testing of plasma oxalate suggests that there is some degree of variability, and this is something that we will learn as the study evolves for its potential use as a biomarker in further studies. We've had patients exposed up to 12 weeks. This is the longest exposure that we've had to our program, and to date we've identified no safety concerns. As we've committed previously, we expect to present data from this trial, interim data, at a Q2 scientific meeting, and then to bring the final data towards the end of the year. With that, I'll shift to our second program, an ALN-346. This is a program designed to treat patients with gout and moderate to severe chronic kidney disease. As you may know, patients with gout, despite there being multiple approved therapies and therapeutic classes, the gout market is still underserved. The prevalence of gout is increasing, and in patients with severe gout or with complicated gout due to chronic kidney disease, they are either restricted in their dosing or their ability to use other therapeutics because of the disease, as well as the renal function that's required in that patient population. For this, and in order to meet this unmet need, we designed ALN-346. This is a novel urate degrading enzyme which we engineered for specific activity and stability in the GI tract. It will be a gut-restricted enzyme working on this. We've done animal proof of concept studies in a animal mouse model to look at the hyperuricemia and chronic kidney disease. This data was presented at the ACR this past fall, and the abstract was quite well received. As we've talked to KOLs and looked at the opportunity in the setting, while gout has many patients and allopurinol is the lead therapy in this space, we believe that there is significant room for patients with chronic kidney disease or other complicated forms of gout. As I wrap up, this is a slide that we've consistently showed across the year and a half since the company IPO'd. What I think we're most proud of is that we've made significant progress in that we have consistently delivered on our planned milestones associated with drug development, including initiating UREROX-1 around this time last year, initiating UREROX-2, our 2 phase 3 studies at the end of last year, and continuing to advance the 346 program. As we think about the year ahead, we are on track, as I mentioned before, to deliver initial data in 2Q in the 206 study, the basket trial in the most severe population. And realistically, our biggest milestone of the year ahead is the top-line data from UREROX-1, our 301 study, which will read out in the second half of this year. I thank you for your attention.